Ginny LaBelle with Defined Health, and we welcome you to another edition of our Insights Series webinars. Today, Mike Rice, Senior Consultant for Defined Health, will present Changing AML Outcomes via Personalized Medicine, Transforming Cancer Management with Genetic Insight. Defined Health is pleased to bring you these webinar presentations as a way of showcasing how we view and think looking forward about various critically relevant topics for the biopharmaceutical industry. Our viewpoint is the result of our long history of consulting with hundreds of pharma, specialty pharma, biotech, and investor clients. In these engagements, we bring our unique perspective, grown over 30 years of evaluating pipeline and inline opportunities by bridging our knowledge of drug development, business development, and commercial strategy across multiple therapeutic categories and technology platforms. I want to quickly call your attention to two of our upcoming in-person events happening in March of 2013. Next week, Defined Health will present the 24th Annual Cancer Progress Conference in New York. That's being held on March 5th and 6th. You may refer to our website, cancerprogressbydh.com, for further information on this important conference which brings together world-class cancer researchers from academia and industry. Also upcoming is Therapeutic Insight by Defined Health, which will be held at EBD Group's BioEurope Spring Partnering Conference. That will take place in 2 weeks on March 11th through 13th in Barcelona, and Defined Health will be convening a series of panels and sessions focused on proof of relevance, antibiotics, oncology, and CNS orphan diseases at that conference. Please, if you're attending the conference, do book a session with us in the partnering booth, and also mark off your partnering calendars to attend our Therapeutic Insight sessions. To learn more about Defined Health, our capabilities, and our upcoming annual Therapeutic Insight and Cancer Progress conferences, please visit our website at definedhealth.com or send us an email or give us a call, and we'll be happy to tell you more. Couple of housekeeping notes before I turn it over to Mike. The slides will be up on our website shortly. There is a previous version of this talk already up on the website. We will take questions at the end if we have time. If we don't get to them, we'll follow up with you in the next day or so, so send your questions as you think of them. And we hope you enjoy the presentation. Thank you, and I'll turn it over to Mike.
Thank you, Ginny. Hi, everybody. My name is Mike Rice. I'm a senior consultant at Defined Health. I co-lead the oncology practice. I focus mostly on hematological malignancies and benign hematology indications. And what this presentation today is sort of feeding off of a couple presentations that we did last year, one on the broader spectrum of heme disorders, and another one on personalized medicine. So this year we decided we would take one of those topics and focus on how do you apply what is coming out of the genome sequencing efforts and genetic association studies to try to tackle problems in a very intractable disease such as AML. And we hope to repeat this across some other tumor types as well in upcoming seminars. And what I'm presenting today is very similar to what I had presented in November. So I know a lot of you couldn't have made it then because of the reschedule from Hurricane Sandy. So if you did attend that webinar, this won't be very similar, but I did add some content and focused it a little bit. So. Okay. So I just wanted to start broadly just talking about the oncology market and some of the challenges and opportunities that we see in moving new therapies forward. So the oncology market now has become the largest therapy area, outgrowing the largest areas such as cardiovascular disease and neurology. And we see a lot of future growth remaining in that. And one of the reasons that cardiovascular disease has taken the dip is due to a generic standard of care that is perceived to be somewhat effective, even though there is still considerable cost in terms of mortality and morbidity to patients as well as cost to healthcare systems. But oncology is sort of the second runner in that. But even though we have a fair number of generic chemotherapeutic agents and some of the target agents are soon becoming genericized as well, there's still perceived to be considerable unmet need, and that the standard of care, even with those cheaper generic agents, isn't sufficient. And so there's a need for improved therapies, and we see this area supporting value-based pricing in the future. So that said, the growth really is being driven by monoclonal antibodies and targeted small molecules, as many of the older foundational chemotherapies and cytotoxics have become genericized, and we see considerable growth coming up in the future. So if we compare solid tumors and hematological malignancies, HemOnc has been a pioneer for approving biomarker-driven and biologically based drugs. If we just sort of compare the newly introduced drugs over the past decade or so, we see that even though heme malignancies are much smaller in number as far as new incidents, they have supported a similar number of new product approvals in with targeted agents, and that is translated into roughly half of the revenues coming from the top 10 blockbuster drugs are now, because of these heme malignancies and some of the progress and transformation that we've seen there. And it's interesting to see, because heme malignancies that I mentioned only represent about 10% of the newly diagnosed cases and deaths each year, yet they're contributing to 25% of the overall oncology market, which is now estimated around $80 billion. And heme malignancies represent $20 billion of that, so it's only 10% of the new patient population that is driving that. And the big areas that are contributing to that are the multiple myeloma area, CML, and NHL, particularly through a few drug classes that have become quite successful in those spaces. The rest of the heme malignancies are still considered mostly, unmet needs, and in particular, what we're going to talk about today, AML has almost no market because the mainstay of therapy is completely genericized. However, there is still considerable unmet need for moving new therapies in there. And looking to the future, we see heme malignancies being a growing space, mostly driven by those targeted small molecule cytostatics and antibodies and immunotherapies that we mentioned prior. But we also see an area for incremental innovation of improvements of existing cytotoxic drug classes, the nucleoside agents, as well as anthracyclines and things like that. Okay. So if we look at the pipeline, we see many more agents that are in early stage development geared towards solid tumors, and much fewer programs in the heme malignancy side, and much of that probably tracks the epidemiology and the perceived opportunity there. However, what we see is when we follow programs, how they are progressing through the clinic, we see a much lower attrition rate, or many programs surviving to later stages of therapy, as many of the solid tumor programs either get repositioned or fail to prove concept. And indeed, if you look back at historical attrition rates in the heme malignancies versus solid tumors, you see a much higher success rate in areas such as lymphoma and leukemia, multiple myeloma, compared to some of the the larger big 5 tumor types. And much of this is driven by, one, understanding a better understanding of the molecular drivers of the disease, and having better understanding the scientific basis and clinical translation of targets in heme malignancies, And if we look at the pipeline and look at how many programs or clinical trials are incorporating biomarker strategies, with companion diagnostics or without, we see that roughly 80% of the oncology pipeline had some sort of a biomarker associated with it, and we see many more companion diagnostics also being incorporated very early on into drug development programs. So what we see is a potentially a very changing space with the incorporation of genomic biomarkers and other biomarkers into the market. And since there are so many more solid tumor programs in the clinic, and many of them have biomarkers associated with them, we probably see the biggest impact to companion diagnostics and biomarkers probably in the solid tumor side, which adds a lot of challenges to how clinicians are going to figure out how to use all these new agents in proper patient segments and being able to implement the proper diagnostic tools and make them available to them. Okay. And also what we see, this figure's a little bit old because some of these programs have been approved, such as crizotinib and the Plexicon drug. But we see that when genetic evidence is used early on to found the scientific basis for drug development programs, they tend to be shorter timelines of getting a product from a scientific discovery to clinical translation to actually getting a drug approved. And we see many programs pursuing these avenues more increasingly. And even in very, very rare tumor types defined by these genetic markers. Okay? So, all right, so that's it. So that's where the oncology market and maybe where it's going. The other side, I just want to touch on this very briefly, is that there's been acceleration in molecular tools and being able to provide genetic information not only to understand new targets and assist with translation of new therapies, but also potentially making them clinically available to clinicians to be able to make treatment decisions. And one of the drivers behind that is just the tremendous advance in technology, not only making more robust technology that is more reliable and easier to use, but also driving down the cost and increasing throughput while maintaining fidelity of those platforms. So we've seen the technology go from the old page sequencing, gel sequencing, Sanger sequencing to capillary-based, which took a little bit of labor out of the work, but essentially had the same sort of approach, to microarray-based technologies that now made things, you can make things massively paralleled. However, you had short reads and potentially increased errors in the output, to what's going on now in solid phase sequencing and the new nanopore technology that could really drive down the cost and increase throughput while maintaining a very high fidelity and be able to do things that we haven't been able to do with older sequencing technologies. And just a note on the cost, so we've now broke the $10,000 per genome cost, and we see this actually in some cases now coming down to about $1,000, and with the new nanopore technology it could even go into millions. Okay, so just a quick view of what's been going on. So Illumina had really transformed and reduced the cost and improved the throughput of sequencing through the use of fluorescent dyes. Ion Torrent now has a potentially much cheaper platform that's being adopted based on using protons instead of fluorescent tags. And the nanopore technology now is coming on that could actually drive costs down to even below $1,000 per genome in a very rapid time. And there's a whole host of other technologies behind that, mainly aiming to address the speed and throughput of the technology and being able to make something more diagnostically useful at the point of care. And here's just a example of one of those devices. Here's a very small, inexpensive device that a physician office or diagnostic lab could buy for about $900 right now. That has very high fidelity, has a good throughput, and could be actually implemented at point of care in a physician's office. So this is something that could be really game-changing, taking genetic technologies out of reference labs and moving them into point of care. The other thing is that this progress in sequencing technology is really rapidly putting out genetic information that, in some cases, in some tumors, we now know, we think we know at least, all of the mutations that might be associated with the disease, and we'll get into this in a few minutes. But there's really a worldwide effort looking at multiple tumor types and outside of oncology and many other diseases as well, and being able to translate that into things that are useful for drug development, new molecular targets, and ways to enable prognostication of patients and for treatment decisions. So the other thing is that we've been focused on the coding genome for so long. The ENCODE program now has released the sequence and potential implications of the non-coding genome, and we find out that it's actually very active in gene regulation and regulating transcription and replication and things like that, things that are vital to the survival of a cancer cell, among other things. So we're now learning that the issues are much more complicated than what we thought. It's not just kinases and signal transduction pathways, but there's other epigenetic influences of cancer progression and survival and drug resistance, and much of this information is being incorporated. And in fact, we'll get into what we're learning about epigenetics, particularly in AML, in a couple minutes. But right now, with all this information, we're sort of up to our eyeballs in information. Clinicians are very confused about what it all means and are fearful of the challenges it might present to their practices of being able to implement it. And researchers still are understanding what are the genetic maps, what is the actual influence of the disease, what are drivers of disease, what are just influencers of disease, and which ones are sort of irrelevant in noise. In that front, this is the most challenging part, and it's lagging our identification of gene variation, but actually being able to translate that into a clinical trait. And even though this is progressing rapidly, we see it sort of advancing as sort of a indication or patient segment by patient segment as we learn more about this more laborious aspect of genomics. Okay. So the recent progress in the heme malignancies has been really largely driven by long-known biomarkers implemented in blood cancers. However, many cancers such as the AML have been really refractory to many of these traditional drug development approaches. Just a brief overview of heme malignancies. So they're type of cancers that affect the blood, bone marrow, and lymph nodes. We separate them into what is the lymphoid malignancies versus the myeloid malignancies. The lymphoid lineage turns into lymphomas and lymphocytic leukemias and myelomas, while the myeloid lineage turns into acute myelogenous leukemia, myelodysplastic syndromes, and myeloproliferative neoplasms and other malignancies. Today we're going to focus a little bit more in the myeloid side of things. And if you look across the spectrum of all these small subsets of the heme malignancies, you see a very wide range of unmet need in terms of survival and age of onset. And just, many of these slower progressing tumors such as CLL and some of the indolent lymphomas and even some of the myelodysplastic syndromes are somewhat slowly progressing. They are diagnosed late in life, usually in the the mid-60s, the 70s, and in some cases they're better managed by watching the disease course and seeing if any intervention is needed at all. So in some respect, they have different challenges in developing drugs for the slower progressing tumors in older patients. We have other ones that affect predominantly pediatrics, such as ALL. Here we show sort of a rolled up ball of both the elderly ALL as well as childhood leukemia. So actually in children we do much better, where we have about a 95% survival rate, while adults have a very poor prognosis. So it just sort of rolls the average in here. Today what we're going to talk about mostly is about AML, which again is a disease primarily of the elderly. We know that younger patients, again, have a much better prognosis than older patients, some of it having to do with the disease itself and some of the different molecular drivers of the disease, and some of it having to do with the patient themselves and being able to tolerate the regimens or intense chemotherapies that younger patients can tolerate, but older patients just don't have the reserves for. Just taking a few cases, looking at some wins that we've had in heme malignancies. So if we take the case of CML, so this is something where I say, we've developed a therapy for an old biomarker, the Philadelphia chromosome, characterized by the BCR-ABL fusion, has been one of the older biomarkers that have been known in heme malignancies dating back to the 60s. And it was, not until we understood molecular genetics and were able to map and identify, why this particular kinase was driving CML, that we're able to develop a small molecule drug, Gleevec, that I'm sure everybody knows very well, which has really transformed the disease from something that, you had a 50-50 chance of making it through 5 years to something now that we have patients on chronic therapy for decades at this point, with minimal long-term effects from the therapy, and it really changed the prognosis, as well as a whole host of second-generation ABL kinase inhibitors that are in some cases even better than Gleevec. So we have a bunch of different lines of therapies if the patient indeed does develop any sort of resistance. Another one related to the topic we're talking about today is APL. APL is a subset of AML, which is also known as M3 AML. It's characterized by a fusion between chromosome 17 and 15 between the retinoic acid receptor and PML. And we've been able to identify sort of serendipitously a nice response to drugs that were originally tried in China, all-trans retinoic acid and arsenic trioxide, that this sort of more poor prognostic subgroup of AML, now through advancement of also, even replacing chemotherapy in some places with these two agents, have been able to really transform this one particular subset of AML into something that has a very good prognosis today. And the last thing has been pediatric leukemia, ALL, has been incrementally advanced sort of trial by trial, cohort by cohort over the last, since the 60s, that has really incrementally, step by step, almost made this a curable disease for at least 95% of children today. And we have a lot of other, salvage strategies for the ones that do indeed relapse. So the point here is that, progress has been made both from, understanding the biology, being able to develop targeted therapies, but also it's been somewhat serendipitous, where, a clinical observation was later found to have a genetic target, and a rationale for the molecular driver of the disease, to another disease that was just incrementally advanced with just trial and error through the years. So now I'd like to focus on AML. So AML now, I'll get into some of the aspects of it, just the basics of it. So AML is a life-threatening disease in which hematopoietic progenitors of the myeloid lineage become cancerous and become dedifferentiated, and they don't go through normal apoptosis, and essentially they squeeze out the normal bone marrow and inhibit production of normal neutrophils, basophils, and thrombocytes. And AML is one of the more prevalent heme malignancies in older patients. New incidence is about 13,000 cases in the U.S., and the mortality rate is very, very high. Even in patients that do achieve a complete remission, it often relapses, and the remissions are not very long. And for an older patient, an elderly patient with AML, the prognosis is probably 5 to 10 months for most individuals. And so we're looking at a very different situation than the pediatric leukemia that we talked about, where you can tolerate intense chemotherapies. Here, a lot of the progress has been limited by the patient not being able to take curative therapies and not being good candidates for transplant. So even a long time ago, we realized that AML is not one disease. It's a bunch of different diseases, and we classified them to these 8 subsets right now. And based on the cell morphology and some of the characteristics of the disease, we've been able to pick out ones that have potentially better prognosis and ones that have poor prognosis, and we've adapted clinical trials strategies and treatment strategies to be able to risk adapt patients based on their expected prognosis. But a lot of the even currently used prognostic information are really clinical presentation. What is that AML subclassification? Is it one that has associated with a better outcome or a poorer outcome? What is the age of the patient, general health? What is their comorbidities and performance status? How they expect it to tolerate the backbone chemotherapeutic agent 7+ regimen 7+3 in AML? Have they had another malignancy that might have progressed to AML, such as myelodysplastic syndromes, or a secondary AML from treatment of other solid tumors and other malignancies? And how does that influence what the patient's outcome is expected to be? We then have added molecular cytogenetic information on top of that, things that we could see based on gross chromosomal translocations and aberrations, and found them to be even better independent risk predictors than some of the other clinical criteria, and so they rapidly were incorporated into the treatment stratification of patients. And so we've developed strategies. We know what to do with the poor prognosis patients. We know what to do with the more favorable risk groups, but the big challenge is what to do with the intermediate group, and we tend to be, currently probably over-treating a lot of people just because you don't know who is likely to have a better response to therapy and who's likely to develop drug resistance. And so on top of that, we've been going finer and finer, looking at molecular markers, and so on top of cytogenetics, we've added molecular genetics and point mutations and other sorts of microlesions to understand who's likely to, within that group, how can we better define treatment risk. Okay, and indeed that has been a viable strategy, and we've been able to take that intermediate group and push some of them based on some of these molecular markers into the more unfavorable group, and some of them more into the good prognostic arm. and indeed improve treatment algorithms and treatment outcomes. And this has been incorporated into many of the cooperative group trials and the NCCN recommendations for how patients should be stratified and risk adjusted. So that said, so we've learned a lot about the disease. We've learned a lot about the genetics and biomarkers of it, and we can understand somewhat who might make it through treatment better than others. However, the treatment approach has not really changed much in over 30 years. So I mentioned that this is a very simplified version of what's going on. We have much more detailed treatment algorithms that we use on our project work, but just for briefly to walk through it, it really has three components. Remission induction therapy. The mainstay of that is cytarabine plus an anthracycline like daunorubicin. The treatment regimens referred to as 7+3 because of that. And this has become the foundational treatment approach for induction therapy. And the goal there is to get a patient into complete remission, which right now is defined by less than 5% blasts, with so much of a crude endpoint with some of the molecular minimal residual disease techniques and tools that we have available to us that we can look at in much deeper response to the therapy. So, and indeed these are being used as one of the risk prognosticators in treatment of AML. An early response to therapy and a deep remission tends to translate to better outcomes of overall treatment. The next step is, and one of the considerations there is really is age and performance status. Is a patient able to tolerate that 7+3 treatment, or are they considered poor candidates for that and could only go through lower dose chemotherapies that have much more poor prognosis or outcomes associated with them? So what we know is that after induction therapy is that if there is an additional treatment, that patients usually relapse very rapidly. We've developed strategies for post-remission consolidation, and depending on your risk assessment, you're now stratified for either more intensive therapy or less intensive therapy based on your this whole collection of risk factors that we've collected through the first week of therapy and your clinical presentation. And if you're considered high risk, then you're a transplant candidate. You're likely to go on to a allogeneic transplant. If you're a lower risk, you're liable to go for just high dose cytarabine treatment or one of the consolidation regimens that are being used, and and then consider that you have a good chance of being cured. Particularly younger patients have a chance of being much higher chance of being cured based on that strategy alone. So patients are just watched during remission to see if they are have a relapse. So then, older patients often are not good candidates for intensive consolidation therapy. So they also go on to some low-dose chemotherapy maintenance regimens, but maintenance therapy in general does not have a good role in the management of AML like it does in ALL and APL and some other malignancies. If indeed a patient does relapse, so if they're not a good candidate for a bone marrow transplant, there are not a lot of good options right now of salvage therapy. We have, if it was a long remission, we can try the 7+3 induction regimen again. But aside from that, there are not very good monotherapy agents that are available right now. And your best hope is really to go on to a clinical trial protocol looking at some investigational agents. Okay, so AML, so if we look across the heme malignancies, so regardless of the advances and the understanding of molecular biology, AML remains, among the highest unmet need in cancers in terms of its mortality and the size of the patient population that is affected. And so what we would like to do is really being able to kind of move this ball, this purple ball, from the left-hand side, move it all the way over into where CML is today. And that's the challenge that we have before us right now. And again, just to emphasize the challenge that we have is that it's not just the understanding of the disease itself, it's also understanding how do you develop regimens that an older patient is not likely to die from the treatment themselves than they are the disease. And that's been some of the limitations of developing new therapies for AML. And as you see, there's a very different outcome for a younger patient, particularly a younger patient that has a bone marrow transplant option where they may have an 80% chance of getting a cure, to an older patient, above 60 that is not a transplant candidate that now has only a 20% chance of getting a cure. And unfortunately, since half of the new incidence of AML is over the age of 68, this is where half the patient population falls. However, so despite the seemingly low benchmark, many agents have failed to improve the standard of care. And some of it may be due to the development strategy. If we look at things like Bayon's loromustine and Genzyme's clofarabine that ran trials, single-arm trials, comparing against historical controls, that the regulatory agencies had pushed back on this development strategy without proving overall survival, comparing against something, at least physician's choice or best supportive care or something like that, like hydroxyurea. So these agents were not able to get registered, even though some things like clofarabine still get used as evidence-based medicine anyway, particularly in areas like bridging the transplant and whatnot. Many other programs also looking at elderly AML had also failed to either show improvements in inducing CRs, complete remissions, when added into to induction or consolidation regimens. Others ones weren't able to show an improvement in survival as monotherapies, and in some cases, even things that we've looked at like FLT3 inhibitors showed a nice CR response. However, they were very fleeting and did not impact overall survival, even though we're sort of revisiting that strategy today. So regardless of this unmet need and low benchmark for getting a new product to show clinical benefit, we haven't been able to do it yet. Aside from Dacogen's recent approval in Europe, which was not approved here in the FDA, but Europe has seen the expanded analysis of that data set and granted approval there. And it's enough, the market right now, so the AML market, as I mentioned earlier, is almost non-existent. But just, an incremental advancement in the space is likely to drive, significant sales in AML. On the horizon, we see near-term impact. Agents with near-term impact might be those sort of incremental tweaks to foundational therapies like vosaroxins, being able to improve upon anthracycline class by avoiding multiple drug resistance and some other limitations of the anthracyclines, such as cardiotoxicity. We see L-cytarabine from Clavis potentially being able to improve upon foundational chemotherapeutic agent cytarabine, being able to avoid resistant mechanisms to cytarabine, and then cyclocel sapecitabine has actually a very nice value proposition for elderly patients as being a tolerable oral agent that might be able to be used with hypomethylating agents. So that's it. This is what we see in the near-term horizon, but there is an opportunity for both incremental advancement in the space as well as potentially new transformative therapies. Okay. So academia, biotech, pharma, philanthropic groups, everybody is kind of working together and figuring out how do they, leverage the information that's coming out of genome sequencing and other biomarkers, and how they improve the use of existing therapies and how they incorporate new classes of drugs. So AML is actually a fairly active area of development. It's almost 1/5 of heme malignancies, and if we just look at the clinical pipeline, there are many, many more agents in discovery and preclinical. They haven't entered clinic yet. So there are a number of shots on goal right now. If we look at the AML diversity across the mechanisms, it's probably one of the most diverse pipelines, aside from maybe non-Hodgkin's lymphoma, where we have multiple treatment strategies in clinical development, not let alone what's coming out of discovery right now. And if we look across, the clinical trial continuum, we see many things in phase 1. Many of the more innovative things are there. In phase 3, we see again those incremental improved cytotoxics and hypomethylating agents, epigenetic drugs, and also what we see is a shift by patient segments. So if you look down the left-hand side of it, what we tried to do is divide the pipeline by both the mechanisms, the stage of testing, as well as which patient segment they're being studied in. And what you see is that even though many drugs start out in phase 1 in relapse refractory setting, because of the unmet need and the size of the patient population and the ability to potentially test your drug in de novo untreated patients, we see many of the agents are progressing for elderly AML and not really being tested in relapse refractory. And so this has become also a crowded space, even though I think there's still room for some other options, particularly things that are novel and tolerable. Okay, so I'm just going to jump through a couple of these, but because I really want to spend most of the time on what's coming out of the whole genome sequencing efforts and what is the promise of genomics in transforming outcomes for AML patients. So AML, as I mentioned earlier, if you can see this up here, it's all the way at the top, leads the pack. We now have, over 150 AML genomes sequenced, and that is actually accelerating quite quickly with some of the efforts that are ongoing internationally. Sort of neck and neck to that is breast cancer. But the first AML genome was only sequenced and published in 2008, but now we've been able to sort of, really trace advancement of AML through different patient segments, whether it's coming from a myelodysplastic syndrome and now transforming into AML, whether it's a relapsed AML patient, and being able to find out what's going on in the changes in mutational status as the disease progresses from one form to another. And indeed, the publication in AML incorporating genomics and trying to understand how do we translate and what are the implications all these different mutations have rapidly advanced since those genomes have been published. So here's a couple examples of some of the seminal publications that came out in AML genomics. The first one published last year, beginning of last year, January 2012, looked at understanding what are the mutational drivers of AML relapse. So what they did is they followed patients prior to relapse and at relapse to understand what is the differences in clonality of the AML clone. Was it the foundational clone, founder clone that survived cytotoxic therapy and then began to regrow at relapse? Was it a new acquired mutation, somatic mutation through that process that drove the relapse? And what are the implications of certain gene mutations versus others in this sort of continuum of disease progression? And they found a lot of very interesting things. In many cases it was, and right now that there's probably not enough data set to understand what is the more commonly occurrence, but some cases there were foundational clones that were more refractory to chemotherapy that were able to survive and then cause implant fluence. And other ones, there were other mutations that were acquired, and a subset was responsible for relapse. So we're actually learning a lot about the disease. What makes patients become cured from standard induction and consolidation therapy, and what happens to patients when and what happens to patients when they relapse, and what is the difference between them. Another thing that we looked at was progression from myelodysplastic syndromes and myeloproliferative neoplasms, and to understand what are the differences between the myelodysplasia to outright frank AML. And we were able to find certain things that are common across AML and MDS, and some things are quite different that now are suspected to be interesting influences and targets for new therapies. So indeed, we were able to take some of this information and again incorporate it into clinical management and be able to understand how do you identify some of these biomarkers that make a patient either less or more amenable, or how do you improve patient outcomes based on risk stratifying them based on the biomarker. And what we found out is that there are many that don't benefit from additional chemotherapy, but there are some, such as DNA methyltransferase 3A, NPM1, and MLL translocations that are now, if they're identified earlier on, that they are good candidates for additional cytotoxic therapy, more intensive chemotherapy, or early transplant, as having better outcomes if they're being able to be identified early. So with the existing treatment that we have today, we've been able to improve outcomes for some patients. So if we look at the targets that came out of these whole genome sequencing efforts, we see a lot of the well-known markers in AML and MDS, but we also see a lot of novel markers that only came out from us being able to fully sequence everything and look at the point mutations driving the disease. Or the challenge right now is to try to figure out which one of those are actually driving the disease and which one might be sort of contributing to the disease or just riding along. And and this is one of the models of how AML might be progressing. And what we really need to do is find out what is that real driver that is the best the target for intervention so that we can sort of prioritize drug development programs going forward. So just to focus on one aspect of it, of all these targets that come out, so we have observed that a large number of these variants in myeloid malignancies appear to be genes whose functions are involved in epigenetic regulation and gene transcription. So many of these are not traditional targets for small molecule kinase inhibitors, even though some are. Some of them have been what we perceive to be, undruggable or hard to drug targets, and some of it are just things that we're just understanding the biology and how they're contributing to mutagenesis. So these roughly break down into three categories that we consider. The epigenetic writers, these are the histone methyltransferases and some other things that are contributing to influencing gene silencing. The erasers, which are the histone demethylases and some other enzymes. And then the areas, the ones that are just really reading the changing of the chromatin, these are the readers, the binders, the chromatin, which now we're developing ways to also make them druggable as well. And if we look across these classes, so we see pretty rapid progress in what's going on. So we've had DNA methyltransferase inhibitors on the market, such as azacitidine and cytidine for quite a while. But when we look at these new, more targeted epigenetic agents, they're just starting to come into clinical development now, and indeed many of them are still in basic studies and early preclinical development, even as some of them are now entering phase 1. And we'll get into a couple of these programs. Again, if we look across the drug development pipeline and find out where there are drug development programs, so if we look at this, if we follow the mutated gene and look at development projects that are targeted at them, there are many programs actually that we can find in the databases, but most of them are being developed in other therapy areas or indications outside of AML. If we look particularly at AML, there are several programs now that are in preclinical development or even have recently entered phase 1 development. Some of them are directly impacting these targets, but some of them are also indirectly impacting them as well. Okay, so Epigenetics, so we found that there are some targets that have been rapidly translated, at least in preclinical models, from what came out of the genome sequencing efforts, and we have sort of looked at and identified things that have gain of function and contribute to aberrant chromatin remodeling. We've looked at things that also have loss of function, and consider all these drug types, even though some of them are more easily druggable than others. And because of the broad nature, and these are targets that are really actually well conserved through nature from yeast and Drosophila, they're very vital and broad spectrum components of biology. And so they really broad implications, not just to AML, but to other diseases as well, which makes a lot of opportunity for it, but also makes potentially some challenges of, how do you prioritize things going forward, and how do you understand the long-term or the off-target effects of such drug development. So here's a couple of programs. So if we took the isocitrate dehydrogenase 1 and 2 programs, it was only in 2009 where they were first discovered. They were correlated to AML. And Agios Pharmaceuticals was awarded a grant to study these as cancer metabolism targets and potentially try to translate them into therapies. This led to a very early, fairly high-value deal with Celgene around developing IDH1. And as well as they've developed very early on in these drug development strategies companion diagnostics through diagnostics providers such as Qiagen. Another target is EZH2, enhancer zest homolog 2, which is also very highly conserved methyltransferase. This is also very rapidly moving by basically 2 companies that have really started this and progressed very, very quickly. Epizyme and Constellation. Epizyme looks like they're about ready to enter the clinic with their program. Both have good strategic partners in advancing the programs, as well as, again, collaborations with diagnostic providers to develop companion diagnostics for these genetically defined tumors. And in AML, it's probably the most advanced genetically defined therapy that is in the clinic now is Epizyme's .1L inhibitor, which also came out of the whole genome sequencing studies, and it indirectly affects the MLL translocation AML and other acute leukemias such as ALL. And this has certain other challenges in developing is how do you move forward a program that is targeting a very rare subset of acute leukemias that is not just one disease. There are several diseases, but it's also quite rare and predominantly affects children more than elderly people. So you have different development issues with this. However, this is something that we look at. They might have quite good promise and prospects in moving a therapy very quickly for these patients, if indeed they can conduct a proper clinical trial there. And again, it also is driven a fair amount of investment in this space, and this would be a good win for validating approach of modifying histone-methyltransferases. Okay, and then beyond this, we have many other things that are being rapidly translated. Again, here are the bromodomain inhibitors and companies that are working in this space. Constellation, again, has a nice program that they're moving forward. GSK has one that they're developing in collaboration, and Tensha Therapeutics also was a very true investment very early on in developing these therapies as well. Okay, so I know I rushed through that. If anybody has any questions, you can send me an email, or we can take it offline. But so just to sort of wrap up in the last couple minutes is that, so in the not too distant future, oncologists will have access to an increasingly complex data set, and the challenge is going to be, how do we make this clinically useful? How do we transform patient management from the typical clinical presentation and an empiric information that a physician uses to make a treatment choice to one guided by a complex set of biomarkers, likely needing some sort of computer-assisted predictive algorithm to personalize cocktails of therapies for patients that have any of those gene targets, the mutations, but also combinations of them that you may need a mix of different inhibitors that to make it most effective. And so that's going to be the biggest challenge is how do we go from this current environment on the left-hand side of this figure to this in future where we now have standard molecular profiling. We have real-time PCR being used, microarray analysis, other things like glycomics and other things beyond genomics that can really take a very complex data set, and so that we understand the disease of the patient more than right now just treating the patient, where we understand the patient more than the disease. And indeed, we hear from academic institutions that strategies to do whole genome sequencing, at least of the regions that are most implicated prognostically in AML, now are becoming mandatory as patients walk in the door. Even though this is sort of taking off center by center, it'll take some time to implement. But the take-home message, I think, is that this isn't the genomics of the 90s. This is sort of a different set of tools that we have, and costs of making this broadly applicable. And even though there are many challenges ahead, and a lot of things I presented were probably very optimistic, but even though we have a lot of challenges ahead, probably AML and some of these spaces are the areas where we could see the first movement in the space, and hopefully there are improvements for outcomes right around the corner. So that wraps up for today. So thank you for everybody for hanging in there. I just wanted to leave you with one last thing and remind you that next week is our Cancer Progress meeting, and if you want to hear the experts talk about improving AML with personalized medicine, we formed a very, very nice panel that is going to bring a lot of expertise and expert opinion into, what are the opportunity and challenges of moving things, new therapies through in space, not just new therapies, but new prognostic tools and things to aid treatment decisions. So we have Brian Drucker will be presenting and discussing, how do we actually understand the whole pathway and how do we, enable new drug development rapidly in the space. We have Eric Hedrick from Epizyme, which will talk on some of the epigenetic programs that we just discussed. Omar Abdel Wahab, who has been an author in many of the genetic association studies from myelodysplastic syndromes and AML, and we'll talk about the state of the art, what's new and exciting that's coming out of those efforts. And then we have Nick Sartlis and Scott Biller, Nick Sartlis from Insight, Scott Biller from Agios. They're also going to talk about, how do we move these new epigenetic agents and targeted therapies and tumor metabolism mechanisms forward, and what sort of challenges do they see in, running these small clinical trials, and even though there's a lot of opportunity and a lot of optimism, there's still a lot of hurdles to overcome to really make this a reality. So that's it for me, and thank you.
Thank you, Mike. That was great. We look forward to any questions or follow-up discussion anyone might like to have, and thank you for joining us. And we will we'll take a breather from our Insight Series briefings in March and start them up again in April. Thank you.